Hi everybody, in this podcast we're going to look at the proper way we do a standard biological drawing, and this is used for when we're recording what we view when we look at specimens under the microscope. So grab your lab book because these notes are going right in there. What I'm going to do first is walk you through the 6 steps that you can record down, and then we will do a demonstration together on how we do this. So the first step when you're recording what you see is to draw the field of view. That's literally the circular area you look at when you look under the microscope. If you remember, you look through and there's this big open area, it's circular because that's the shape of the lens. The second step is to title what you're drawing. Make sure that you have a title that reflects what you're actually looking at. Okay, so nothing like unknown cell. Okay, not okay. All right, you would need to either title it with what it is as a cell structure, or here's a tip, look at the slide, because the slide actually will have information, especially if it's a prepared slide. If it's one you've made, then you ought to know what you've put on it. So make sure that the title reflects literally what you're looking at. The next part is to record the total magnification of what it is that you're seeing so you can get a sense of its relative size. And this is a very easy way to do this. You take the eyepiece magnification and you multiply it by your objective lens. So if I'm looking at something that's under low power, I'll call it LP, okay, my eyepiece, and our eyepieces for our microscopes are all 10x. Sometimes they might be different, so you have to pay attention to what the eyepiece is. And all I do is multiply that by the low power objective, which is also 10x, and voila, the total magnification is 100 times, okay, the size of what it really is. If it's high power, it's a 40x objective lens, eyepiece still 10, and your total magnification is 400x. So that's what you need to do. And the only thing you actually need to write down is your total magnification. So to do that, you just have to pay attention to what the eyepiece magnification is and what the objective lens magnification is. Once you have those down, then the next thing is to draw the object to scale as best you can. All right, so that's something to keep in mind. If it is, taking up half of the field of view, it should only be drawn as half the field of view. And like I said, after I go through just sort of the overview of these steps, then I'll do a demo with you that you can also put in your lab book as an example. So you draw the object to scale. Okay, easy. And for those of you that are a little fearful of drawing, trust me, I'm not going to be looking at your artistic ability. I'm looking at your ability to try to accurately portray what you see. That's all. Then you're going to label visible structures. This part's critical. And at the very end of this, you'll have a list to write down of all the visible cellular organelles we can see with our microscope, because some we just can't. Our microscopes aren't capable of looking at them. And then the last and probably toughest part is to do a size estimate. And we use micrometers, and the abbreviation for micrometers is the Greek letter mu, which looks like this sort of curvy U, M. That equals a micrometer. And so that's the abbreviation we use. And a micrometer is 1,000th of a millimeter. And we'll be working with this conversion. So if I have 1 millimeter, that equates to 1,000 micrometers. So you take the tiny little lines on your ruler, and if I take the distance between those two lines and divide it into 1,000 increments, I have a micrometer. And that's how we will look at the size of the objects that we view. Okay? So those are the 6 steps. Follow those steps, you'll never miss anything. So let's do a demo. So in this one, we're going to look at a demonstration of how we do our drawing. So I have drawn the field of view already, so I've taken care of step 1, which you don't need to write out step 1. I'm just pointing that out to you. Each of your lab drawers will have a petri dish. You want to try to make a very nice, circle so that's not all over the place. So there's that. The second step is for me to title what I'm looking at, all right? And we're going to call this one, let's say we're looking at amoebas, all right? So I would title it amoeba, and generally where I'm putting stuff is about where you should put it. So the title should be towards the top of the field of view. My next step is to do the total magnification. So let's say we're looking at this under high power, which is my 40x lens. All right, and because that's a 40x lens, don't forget your eyepiece. I multiply that by my 10x eyepiece. I put the total magnification underneath, and that's 400x. So cool, I'm already through the third step. Fourth step, okay, is going to be to draw to scale. So I'm going to draw my amoeba. So I'm looking through the microscope and I say, wow, that's amoeba, kind of has this shape to it, and I'm going to do the best I can to draw that. And I'm going to look and I'm going to be like, wow, it's got this thing in it, and it's got a few other little things, but that's primarily what it looks like. So now I've drawn it to scale. Cool. Step 5. And let me label these so you can put them to the steps. So there was step 1, there's step 2, which you don't, again, have to number. There's step 3, and there's step 4, okay? Step 5 would be to label all visible structures. So when I label this, I'm going to label what I see, and I'm going to try to put a line to them. So here is my cell membrane, and you label them right inside, and there's my nucleus and my cytoplasm. And you're going to learn what all these are. All right, so you label only what you can see. And then, so that would be my 5th step. And then the last step is to do a size estimate. And you can do this a number of ways. What I have to do is determine what I'm going to estimate. And in this case, I'm going to bracket off this length. And I'm going to do an estimate of how long my cell is. Okay? And what I'm going to do is jump to the next slide to be able to share with you how we do that, because it involves a little bit of math. So to do a proper size estimate, okay, the first thing we have to do is figure out what power we're looking at. And since we're on high power, I have to figure out first what the diameter is of the field of view. And the lab we're going to do in class, you're going to learn how to measure this out. But I'm going to give you some measurements, okay? So the diameter of the field of view, which is the distance, you know, diameter, right? So I'm going to go with my distance across, okay? If it's low power, the diameter is 1,600 micrometers, okay? And that's at 100x. Now, if I go from low power to high power, I'm going to 400x. Which means when I look at an object and I go to high power, I'm getting closer to it. So my diameter is actually going to shrink, and it shrinks by 4 times. So it gets 4 times smaller because I'm going from 100 to 400x. 4 times, right? So I get 4 times closer to the object, so that means I have to take this 1,600 micrometers, I divide it by the 4 times closer that I get, and my high power field of view diameter is 400 micrometers across. Okay, so again, to do an estimate, you need to know the diameter, so it's really, really important to know what objective you're looking at and what the magnification is. All right, so you just have to convert it. Now that I have my field of view diameter, how I figure out the actual size estimate itself, okay, is I have my diameter. So let's say, you know, my amoeba, I was looking at it under high power. So I take my diameter, and I decide how many of my amoebas I could fit across. And if I go back and look, about 2 amoebas could fit across that diameter end to end. But I don't want to know the size of 2. The size of 2 would be 400 because they fit across. I want to know what the size of 1 is. So I only want 1 out of those 2. Right? I only want to know the size of 1 out of the 2 that could fit across. So this number here is important because that's how many fit across. Right? So if I take 400 and I multiply it by 1 out of 2, or 1/2, that means the size of my amoeba, I'm kind of running out of space, so I'm going to have to do it sideways here. The size of my amoeba was 200 micrometers. Okay? And I would add that to the little bracket I had put back there. All right? So that's how you do a size estimate. All right? And we will practice this. I'll go over it with you guys again. Okay? So just so that you have that down, that's how you're going to do it. So the keys to doing an estimate, you've got to know what the magnification is, so you know your diameter and how many could fit across. So if you know your diameter and how many could fit across, you can find out the estimate, again, rough idea size of what it is that you're looking at. So here is our list of visible organelles. Okay, these are the things we can see. All right, we can see the cell membrane. We can see the cell wall. We can see cytoplasm, which is kind of like the goo everything floats in. We'll be able to see nuclei, or the nucleus of a cell at times. We will be able to see chloroplasts, which are found in plant cells and photosynthetic cells. We can see vacuoles. We will be able to see cilia, okay, which is fuzziness outside. That's how they move. We will be able to see, during cell reproduction, when a cell divides, we'll be able to see spindle fibers, and we'll be able to even see chromosomes sometimes. But spindle fibers and chromosomes, these are only when the cell is dividing. Okay, that is what we can see. Oh, we can also see flagella. Okay, those are the things that we'll be able to see when we look at our organelles. All right, so there you have it, how we do standard biological drawings. And we will practice with this a ton in the lab. We'll have a bunch of labs where we'll be working with this. So take care, and we will see you in the lab when we are practicing all of this. Have a great night, guys, and we'll talk to you later.